Hello students, welcome back to the channel. This is a part 3 of the reproduction in flowering plant. In this video, we are going to discuss the anther wall. You know very well in the previous video we have studied that archosporial cell divide periclinally to form two outer parietal and inner sporogenous cell. Outer parietal will give rise to wall layer. Which wall layer? Epidermis, endothecium, middle layer and tapetum. So we are going to study now these more in detail. Now see, first layer, outermost layer is epidermis. And in the previous video, we have already studied it is not archosporial in origin. It was already present from the beginning. So first layer is epidermis. And it is single cell and it is not archosporial in origin. But one exception is there. In Arcithobium, you will find it will develop fibrous thickening. And that is why their epidermis is also known as exothecium. So please learn this is an exception. Don't confuse with endothecium. Endothecium is a second layer. This is exothecium, which is in Arcithobium you will find epidermis is developing fibrous thickening to form exothecium. Now let me explain you the wall layer. See, I'm taking this side only of the diagram. Please understand, I'll take only this side, okay? So see here, this is the first layer epidermis. This is epidermis. And then next layer is endothecium. This is endothecium. And endothecium will have thickening in the inner wall and radial wall. Inner wall, this side, and radial wall, this side. Is it clear? So they are having thickening on the radial wall and inner wall, not on the outer wall. Please understand the concept. And this thickening is made up of alpha cellulosic. Please understand, because cellulose fibers are made up of beta glucose. It is alpha cellulosic fiber is present. Clear? And this is present in all the area except area which is weak, which is also known as stomium. Stomium means also known as line of descent. If you see in this diagram, I have left this area as a weak area. This is a line of descent, also known as stomium. Please check your NCERT, you will find it. It is stomium, also known as line of descent. So here, the fibrous thickening is absent. Now, so some places fibrous thickening is absent and these places are known as stomium, which help in the descent. Endothecium is hygroscopic in nature, means it will absorb the moisture, and because of the absorption of moisture, it will go for expansion, and unequal thickening will make the anther wall rupture from the stomium side. So it is having not only fibrous thickening of alpha cellulosic, it is having of callose also. Callose is a chemical. Callose is also a chemical which is also present here. So let me just draw it. This way you can learn callose thickening is also there. So cellulose thickening plus callose thickening is present. Now, few exceptions. Fibrous bands are absent in hydrophytes. Next is middle layer. Middle layer is the one which is nearly 2 to 3 layers. I am drawing right now 3 layers in my diagram. This is middle layer and middle layer is 2 to 3 layers. And this layer will not be seen at the time of the anther maturity. This layer is ephemeral, or you can say it is a short-lived layer. Please understand this layer is 1 to 3 cell thick and it is ephemeral, means it is short-lived and it will not be seen in mature anther. Clear? So outermost epidermis, then endothecium and middle layer. These are the 3 layers which play very important role in protection. They play important role in protection. So outer three layer helps in protection and they also help in the dehiscence. Is it clear? This layer, why we say it is ephemeral? Because it is rich in the food which is used, or you can say it is taken by the tapetum to nourish the developing pollen grain. So it is parenchymatous and food is stored in this layer which is used by the tapetum to nourish the developing pollen grain. Innermost layer is tapetum, and tapetum is the one which is providing nourishment. It is single cell thick. They are having dense cytoplasm. Okay, dense cytoplasm, they will have increased the DNA content. Now, how can they increase the DNA content? Either because of endomitosis, means full mitosis is done inside the nuclear envelope only. Prophase, metaphase, anaphase, everything is done inside nuclear membrane. So at the end of the telophase, instead of seeing two nucleus, you will see only one nucleus is present, but DNA content is doubled. Restitution nuclei is a condition in which they are doing normal mitosis, but at the telophase, instead of making two nuclear envelope, they make only one nuclear envelope. Is it clear? Polytene is the condition in which during S phase, instead of making only one, doubling the one chromatid, this way they are increasing their chromatid number only. So obviously what is going on? DNA content will increase. Is it clear? So polytene means DNA content increase without increasing the number of chromosome. Chromosome is still one, but see number of chromatids have increased, so DNA content has increased. So please understand the concept. They are having dense cytoplasm. They are having increased the DNA content. There will be polyploidy. Obviously, DNA content is increasing, so polyploidy and increase in the number of nuclei also possibility. Clear? Now, two types of tapetum will be there. One tapetum in which it will work like a gland. It will work like a gland, means it will not degenerate immediately. It will absorb the nourishment from the middle layer and it will release into the around the pollen grain, which are going for maturity. Okay, so they are releasing the chemic nourishment after absorbing from the middle layer into the area where sporogenous tissues are going for making the pollen tetrad. One time, and this is working like a gland, so obviously it is intact and gland will do the secretion, so we'll say secretory type of tapetum. Second type of tapetum is amoeboid. Now name is telling me it will show amoeba movement. It means it can move, so it is invasive. It will enter inside the middle layer, take the nourishment from the middle layer and it will release near the mature pollen grain, means the pollen grain which are going for maturity. Plasmodium, and it is primitive type. So such tapetal cell will absorb all the food from middle layer. So middle layer immediately degenerate. First they will store it and finally they will convert all the absorbed food into the spatial granule, that is protoplast bodies. They will make protoplast bodies and finally the innermost layer of tapetum will dissolve and release the protoplast into the cavity of microsporangia so that they can provide nourishment to the developing pollen grain. Clear? Now see, understand, this is your PMC, pollen mother cell. Ploidy is 2N, which has undergone meiosis to form pollen tetrad. This is pollen tetrad. Clear? And pollen tetrad means haploid cell. After meiosis, you know the cell will become haploid. So ploidy is N. They are surrounded by a special type of chemical. That chemical is known as callose. Check in your NCERT, one cover is present around the pollen tetrad. Clear? And that is known as callose. This chemical is callose. Understand it properly. See, now what the tapetal cell will release? Tapetal cell will release callase enzyme so that all the microspores separate. So all these 4 microspores separate. Now these 4 microspores are separating. So first it releases callase enzyme. So I think so this is clear. It releases callase enzyme. Clear? Then what will happen? Tapetal cell will release now Yubish body. they will release Yubish body. And Yubish body are the one which contain sporopollenin. They contain sporopollenin. Let me explain you by taking only one microspore. Please try to understand. I'll take only one microspore. This is the one microspore, and here Yubish bodies are present. This is the way Yubish bodies are present. And what they are doing it, they are making one layer. Check. Here, this area where Yubish bodies are not able to make this layer, this is known as germ pore. Right now, I've drawn in my diagram only one germ pore. Germ pore can be one or it can be two or three. Clear? One germ pore will be seen in monocot, two or three can be seen in dicot. So they are having the Yubish body, and Yubish body are the one which are containing the tapetal cell degenerate to release Yubish body into the pollen sac, and Yubish body, they are the one which contain the sporopollenin. Then tapetal cell will release compatible protein. They will release compatible protein because when the pollen pistil interaction has to start, that time compatible protein will help to know which is the right pollen grain for the stigma. Let me draw this dot as a compatible protein. So this is the compatible protein which is present. Clear? Sporopollenin already explained. Then it is also releasing see the diagram carefully. Tapetal cell will also release pollen kit. And what is pollen kit? Pollen kit is a mixture of carotenoid and lipid. So you can see pollen kit, which is a mixture of carotenoid and lipid. And I'll show pollen kit with the blue color. Okay, I'll draw this way, hashtag this way. You can understand this is the way pollen kit. So when you see pollen grain mature, what you're finding the structure, understand, you will find this is the inner wall. Then you're finding this is outer wall of the pollen grain. I am drawing now two germ pores. Then you're finding it is having compatible protein. Clear? Compatible protein. Then you're finding this is the pollen kit for the entomophily. Okay, for entomophily. So normally each PMC will make only one tetrad. Right now I've shown you one PMC is making only one pollen tetrad, and role of tapetum is to release enzyme. It is releasing Yubish body. It is releasing compatible protein. It is making the pollen kit also, as well as it is responsible for the formation of hormone so that the growth can occur because it's responsible for cell division. Clear? But in some plant like Jostera, some PMC becomes sterile to produce nourishment to the rest of the pollen grain. So I hope so in this video you are comfortable with the anther wall in detail properly. Now let us see the MCQ. If I ask you what is the correct order, the first layer is epidermis. You know very well. First layer is epidermis. Second is endothecium. Third will be middle layer. Fourth will be tapetum, and innermost will be sporogenous tissue. So we have to start first from the fourth. So this is fourth first given. Clear? Then, so this is correct up till here. Then second will be number one. Yes, number one, endothecium. Then third will be middle layer. So answer is coming C option. Now, how many layer of microsporangia wall will perform the function of protection and help in dehiscence? We have studied there are three wall layers, especially epidermis, endothecium, middle layer. They are the one which are responsible for protection and helping in dehiscence. So three wall layers. Is it clear? So I hope so you're getting the answer. Now this is the Venn diagram given to you. It is multinucleate. It nourishes. It is having dense cytoplasm. So what is this? This is your tapetum, because you know nourishment word is given for the tapetum. The answer is tapetum. So I hope so in this video, anther wall, all the topic in detail might be clear to you. This video might be helpful for you. And in the next video, we are going to study about microsporogenesis. Thanks for watching.